Chapter 11 is our Mendelian genetics chapter. This chapter talks about the rules of inheritance. Excuse me, I have a bit of a tickle in my throat today. We start talking about inheritance with meiosis because meiosis produces the cells that are going to be passed on to produce individuals in the next generation. The behavior of chromosomes that we saw and the production of those gametes mirror the inheritance patterns of genes that we see in individuals. Those patterns were first elucidated by Gregor Mendel in the 1850s and 60s. Farmers had known for a very, very long time, for thousands of years, that you could breed two individuals, say a plant or a domesticated animal, and get particular desirable traits, and you could increase the frequency of those desirable traits in your population, and you could select for those traits. But Mendel was the first one to really study this, keep really good records on it, and look for specific patterns in these traits that he was looking at. Mendel was an Austrian monk. He was tending the garden at his abbey, and he took a particular interest in these characteristics in pea plants. Mendel was interested in the way that these pea plants inherited particular traits from their parents. Prior to Mendel, the general idea on inheritance was an idea of blending. In this idea, genetic material sort of mixes in the offspring. So your particular trait that you're looking at is somewhere between the trait that mom and the trait that dad had. So down here on the left, we have a red flower crossed with a white flowered plant. And in the blending pattern of inheritance, you get all offspring with an intermediate form, a pink flower. Mendel was the first one to show the idea of particulate inheritance. In this idea, parents pass on traits that retain their identity in the offspring. So the individual offspring are going to have the same trait as one or both of the parents. And we now know that some of those traits or genes are passed on and can be shuffled into new combinations in meiosis. And that'll be important a little bit later. But particulate inheritance is shown in these pea plants on the right. We have the purple flower and the white flower. When these individuals are crossed, the offspring are either purple or white. You don't get sort of a halfway light purple flower. Those traits remain intact in the offspring. And all offspring are one or the other. And this was the first time that anyone had sort of looked at this and followed these inheritance patterns. We will come back to a type of inheritance called incomplete dominance a bit later that actually looks like blending and is sort of behaving in a blending manner. But when we get there, I will talk about how it's actually different from blending. So one problem with this idea of blending is that if individuals breed with others who have a different version of their gene, this parental generation at the top with the white and the red, we see the next generation is some sort of intermediate between either one of those parents, and eventually that trait gets sort of watered down into this light pink. The problem with this is that it causes a loss of genetic variation. It makes the population identical for a particular trait. And if this is happening for all of the genes or all of the traits in a population, eventually all of the individuals are going to be genetically identical, which we'll see when we get to evolution is bad for a population. On the other hand, particulate inheritance increases genetic variation. We saw that when we were looking at the segregation of alleles in meiosis, where homologous chromosomes separate, and then also in independent assortment, where they line up along the metaphase plate in different combinations to produce different combinations of parental chromosomes. And then we also saw this genetic variation increase when we looked at crossing over. So there's a lot of ways that meiosis increases genetic variation. And the link between genetic variation and particulate inheritance is really important, because if your traits are particulate, if you retain those traits in the parent, you're going to get new combinations of those traits, which increases the genetic variation in a population. In blending, if everybody blends into the same thing, you lose that genetic variation, and then evolution has no way to work on that population. So Mendel worked primarily with pea plants, and he studied a variety of different characters. We now call those characters genes in these pea plants. One important thing to remember about Mendel and his work, though, is all of his work took place before there was an understanding or knowledge of meiosis and chromosomes. And this sort of became a problem when he published his work, because I think that people didn't quite understand what he was saying. They didn't really have a mechanism for inheritance. He was saying that these particles were passed on, but nobody knew what those particles were. And so Mendel's work was actually lost for 40 or 50 years until microscope work and being able to visualize chromosomes and their patterns of movement actually caught up with these ideas that Mendel had. But Mendel spent quite a long time studying these pea plants. He crossed, which means bred, thousands of these plants. We'll look at some of the numbers that he had in just a few minutes. So he had a large population to study, and he kept really good records. And that's really important when you're doing a science experiment. You need records of all of the data that you're collecting. And so he collected data on thousands of plants. He analyzed it. He determined the ratios of offsprings with particular traits, and he was able to put all of that information together and make a couple of really important conclusions that are the foundation of the study of genetics. So even without the knowledge of chromosomes, Mendel was able to describe the patterns or the law of inheritance that we know today still fit. And they fit because they follow the movement of chromosomes during meiosis. Pea plants are really good organisms to study for a few reasons. Organisms that are studied often are called model organisms, and there are a variety of model organisms that are still used quite frequently. Fruit flies, E. coli, yeast, white lab mice, all of those guys are model organisms, and they all have a variety of characteristics that makes them really useful for genetic studies. Currently, pea plants are not really model organisms. They're not really used too much for genetic studies anymore, but they still share some characteristics with organisms that are easy to use for genetics. One is that they have easily studied and identifiable traits. These are traits where they have distinct characteristics between the different versions of the characteristics. So they're real easy to put into groups of, say, purple flowers or white flowers, or round peas or wrinkled peas. They're also small and easy to grow. So Mendel was working in a garden plot in his abbey, so he didn't have unlimited space for these plants. So model organisms in general are usually small, easy to take care of, easy to house, easy to feed, so that they can be easily studied. Another important feature is that they produce a lot of offspring. If you're looking at genetics really requires a large population in order to really see the patterns that are occurring. So large number of offspring is another important feature of these model organisms. They also have short life cycles, so they reproduce very quickly. Not only do they produce a lot of offspring, but they produce a lot of offspring very frequently. So Mendel was looking at characters. These are a feature that varies among individuals. Today we call those genes. So he was looking at the character or the gene for flower color. And then he was also looking at the traits or the variants of those characters. So if we're talking about the flower color character, the two traits that he observed were purple and white. Today we use the term allele, and so I will probably be using gene and allele rather than character and trait because those are the common words. But anything that you read about Mendel will probably say character or trait. So Mendel spent a lot of time crossing his pea plants. In fact, if we go back to this slide, you can see the method for crossing plants. The reason that plants are easy to use is that you can perform fertilization in a very methodical way. So in this case, he would remove the pollen using a paintbrush and then apply it to the stamen of the other plant. So the pollen is the male gamete, the stamen has the ova inside that reproductive feature. And so by removing one or the other, it prevented a plant from self-pollinating, which just means that its own pollen fertilizes its own egg. And so he could either allow them to self-fertilize, or he could cross different traits that he was looking at. Mendel started with true breeding individuals. These are individuals that have been allowed to self-cross for successive generations. And this was really important because it allowed Mendel to know that all individuals from a true breeding cross have the same trait for a particular character, and that they only had information for that trait. So when he started looking at purple flowers, he knew, because he was using true breeding individuals, that though any individuals that would self-pollinate would only ever produce purple flowers. This was important, so he knew what he was starting with. He knew that the only possibility for that purple flower was to produce a purple flower. Same thing with the white. He established these true breeding white flower populations. So he knew that those white flowers always started with white traits. So you can allow a true breeding individual to self-fertilize, or you can do something called hybridization, which is just a fancy word for saying cross-pollinating or breeding these two different true breeding flowers. Up here we have our P generation, that stands for parent generation. So we start by crossing true breeding purple and true breeding white flowers, and we get the F1 generation. F1 stands for filial, so it means the first generation of brothers and sisters or offspring. And if you notice here, he saw only purple flowers. And we'll see why this is important in just a minute. But it appears that that white trait was covered up somehow by that purple trait. So then he allowed that F1 generation to self-pollinate or cross-pollinate. We'll see why it doesn't actually matter which one it is in just a few minutes. And he discovered that in the F2 generation, or in the second generation, he got mostly purple flowers, but he also got some white flowers. So somehow that trait was covered up in the F1 generation, but reappeared in that F2 generation. This is just a summary slide of all of the definitions that I just used. All right, so we come to two important observations from these true breeding parents. The first is that the F1 generation lost one of the traits. This is important for two reasons. One, it's important that that white flower was covered up. It was also important in showing that there was no blending. So Mendel didn't see an intermediate purple trait here. Mendel saw the same purple trait that was in the parent generation. And second, that trait that was lost in the F1 generation reappeared in the F2 generation. So that trait wasn't lost. It was just hidden somewhere in that F1 generation. The other important thing that he observed is that in this F2 generation that was produced from those true breeding parents with one trait covered up in the F1 generation, always displayed a 3-to-1 ratio, meaning 3 of the offspring would have this one trait from the parent that was the one that covered up the other, and one offspring would have the trait that was covered up. Another way to say this is that 3/4 of the offspring were purple and 1/4 was white. In general, we're going to be using ratios, 3-to-1, 2-to-1, etc. But you can also express these as a fraction. So 3/4 were purple, 1/4 were white. Something else to note here is that Mendel was using huge sample sizes. So we have close to 1,000 plants here. So this means that from that F1 generation, he planted 1,000 seeds and got about 700 purple and 225 white. When you have large sample sizes, it reduces the amount of error due to chance. So those huge sample sizes really ensure that he was getting accurate results. So Mendel observed 6 different characters. We see different physical features of these pea plants. Flower colors there at the top. He also looked at seed or pea shape and color. He looked at the pea pod shape and color. He also looked at the position of the flower on the plant, and he looked at the height of the plant. If you notice here, we can see the huge number of plants that he worked with. 6,000 to 2,000 for seed color, 5,000 to 2,000 for seed shape. The other important thing to notice is that all of these display roughly a 3-to-1 ratio. They're not perfect. That is due to chance because genetics is basically chance events. So we expect a little bit of deviation from that 3-to-1 ratio, but that's why those huge numbers in the population were so important. So, as part of his conclusions, he noted that some sort of heritable factor, something was being passed on from one generation to the next, and that white heritable factor wasn't lost. It was simply masked or hidden by the other one. He called that hidden, in this case, white trait, the recessive trait.